I want you to just take a second and imagine a microscopic internet functioning inside your body right now.
Right, and we're not talking about nerves firing electrical impulses here.
Exactly. Not nerves, not blood pumping. A literal wireless communication system. A network where, millions of these tiny, fluid-filled biological flash drives are just constantly being uploaded and downloaded by your cells.
And they are transmitting these incredibly complex, zipped files of genetic instructions across the vast distances of your anatomy.
Which is wild to think about. And welcome to today's deep dive, by the way. Our mission today is to unpack this exact system. We are diving into a really incredible stack of sources today.
Yeah, a fascinating collection of cutting-edge research papers and clinical studies.
All right, focusing on mesenchymal stem cells, extracellular vesicles, and tissue regeneration. Basically, the fascinating world of exosomes.
And we are going to look at how these tiny messengers regulate the physical scaffolding of our bodies and how they can actually reverse severe tissue damage.
And also, because biology is basically one big arms race, how cancer attempts to hijack this whole exosome network. It sounds completely sci-fi, honestly.
I mean, it really does sound like science fiction, but this is rapidly becoming the actual future of regenerative medicine. The sheer volume of data being moved through this exosome system is just staggering.
But it wasn't always viewed this way, right?
No, not at all. For decades, the consensus was that these vesicles were just cellular exhaust. The early literature actually called them apoptotic blebs.
Apoptotic blebs, that sounds like an insult.
It kind of was, in a cellular sense. They thought it was just the debris left over when a cell dies, like a microscopic garbage disposal system.
But the clinical research we have today completely upends that. So before we get into how tissues are repaired or destroyed, we really need to understand these messengers and the environment they actually operate in.
Right, so let us define what an exosome actually is. They're a specific type of extracellular vesicle, and they are incredibly minuscule. We are talking only 40 to 150 nanometers in diameter.
Which is, almost unfathomably small.
It is, but their defining feature is their armor. They are created by a lipid bilayer that encapsulates them.
Which is crucial, right? Because I imagine the bloodstream is a pretty hostile place for loose genetic material.
Oh, incredibly hostile. If a cell just ejected a naked strand of RNA to send a message, roaming enzymes like RNAs would immediately destroy it. They see it as a threat.
So the lipid bilayer acts like this protective transit vehicle. It keeps the cargo, the RNAs, microRNAs, and proteins totally safe from degradation.
Exactly. And originally, like we said, people thought they were garbage bags. But now we know they are vital for paracrine signaling.
Paracrine signaling, that just means local cell-to-cell communication.
Yes. The exosome is secreted, it navigates the space, and docks with the recipient cell. And all of this transit takes place in the extracellular matrix, or the ECM.
Okay, let us unpack the ECM because it comes up constantly in these sources.
Right. The ECM is essentially the structural scaffolding of our tissues. It is made up of things like collagens, elastins, fibronectins.
So is the physical framework, let me try an analogy here. If the ECM is the physical building, or like the steel scaffolding of a city under construction, the exosomes are the foremen.
I like that, yeah.
Right, so they are foremen driving around in armored trucks, the lipid bilayer, and they are handing out flash drives with updated blueprints to the construction workers.
That is a brilliant way to picture it. And what is crazy is that those blueprints can literally change the biology of the recipient cell. They do not just offer suggestions, they rewrite the software on the fly.
Which, if you are listening, just think about how that changes your understanding of your own body. Your cells are not working in isolation at all. They are constantly downloading instructions from each other.
Exactly. But if they are handing out blueprints to construction workers, what happens when the workers receive bad instructions and just go crazy?
They build way too much scaffolding.
Right. And you get fibrosis.
So let us look at how exosomes actually fix this runaway construction. The research highlights oral submucous fibrosis, or OSF.
Yes, OSS is a potentially malignant disorder. It is heavily associated with the cultural practice of chewing areca nuts.
And the areca nut basically triggers this pathway, right? The TGF-beta-1 pathway.
Exactly. That pathway causes the oral fibroblasts, the construction workers in the mouth, to go into hyperproduction. They overproduce insoluble collagen, and they totally stop degrading the old matrix.
So the mouth just fills up with this dense, rigid scar tissue. It is a severe problem. But the solution detailed in the data involves exosomes from a very specific source.
Right. They used human adipose-derived mesenchymal stem cell exosomes, or ADSC exos.
So exosomes derived from fat tissue stem cells.
Yes, from belly fat, essentially. The research shows these specific exosomes average about 58 nanometers, and they are positive for specific markers, CD63 and TSG101.
And those markers are like the identification tags that let them dock with the oral cells, right?
Exactly. They enter the oral fibroblasts and actually reverse the fibrotic damage.
Which is incredible. But how do they physically do it? What is the actual mechanism?
Well, the ADSC exos target the P38 MAPK signaling pathway. They enter the cell, and they do two things. First, they downregulate the genes that are making all that collagen, KO1A1 and CO3A1.
Okay, so they shut down the factories producing the excess scaffolding.
Right. But, crucially, they also upregulate matrix metalloproteinases 1 and 3, MMP-1 and MMP-3.
Okay, here is where it gets really interesting to me. The MMPs are basically the demolition crew, right?
Yes, they are enzymes that chew up the dense matrix proteins.
So the fat-derived exosomes tell the mouth cells to stop building scaffolding and immediately hire a demolition crew to clear the excess rubble.
That is exactly what happens.
That is wild, but how do researchers know for sure that it is the exosomes doing this?
Good question. They proved the pathway was responsible by using a chemical called anisomycin.
Anisomycin, what does that do?
It is a P38 MAPK activator. It artificially forces that runaway construction signal to stay on, and when they used it, it completely reversed the healing effects of the exosomes.
So it proves the exosomes were specifically using that pathway to fix the fibrosis.
Exactly.
Okay, so we just saw MMPs acting as the heroes. They are the demolition crew clearing out bad scar tissue. But any powerful system can go wrong. What happens when the demolition crew goes rogue and will not stop tearing down the building?
Well, then you get diseases like osteoarthritis. In that case, the ECM breakdown is the actual problem.
Right, the architecture of the joint is just dissolving.
Yeah, there's an overproduction of MMP-3. It degrades the cartilage matrix relentlessly.
And that leads to all the pain and swelling.
Right. It leads to synovial hyperplasia and chondrocyte apoptosis, which just means the cells maintaining the joint are dying.
But the sources say they solved this with exosomes from the exact same source, right? Adipose stem cells.
Yes, ASC-derived exosomes.
Okay, wait. How does an exosome from belly fat solve the exact opposite problem? In the mouth, it deployed MMPs, but in the knee, it has to stop them.
It comes down to what is inside the exosome. ASC-derived exosomes actually contain high levels of TIMPs and almost zero MMPs.
TIMPs, tissue inhibitors of metalloproteinases.
Right. TIMPs are the natural antagonists to MMPs. By delivering these TIMPs, the exosomes block the rogue MMP-3 and prevent the cartilage breakdown.
So what does this all mean? It is like a highly intelligent biological pharmacy.
That is a great way to put it.
Right, because in the fibrotic mouth, it dispenses the demolition crew, the MMPs, to clear the blockage. But in the degrading knee joint, it dispenses the riot police, the TIMPs, to stop the demolition crew.
Exactly.
But how does a vesicle from belly fat know exactly what to do in the mouth versus the knee?
Well, it does not have a brain. The intelligence is in the interaction with the local environment. The damaged cells in a fibrotic mouth present entirely different receptors than dying cells in a knee. The recipient cell essentially pulls the specific tools it needs from the exosome's library.
That is just brilliant.
Yeah.
But I mean, any communication network this powerful is vulnerable to being hacked, right?
Absolutely, and that is where the oncology data comes in. Cancer is the ultimate hacker of the exosome network.
So how do diseases use these exosomes against us?
Well, tumors secrete their own corrupted exosomes to manipulate the ECM for their own survival. Specifically, they use them to create a pre-metastatic niche.
A pre-metastatic niche, meaning they prep the soil before they even arrive.
Yes, and this usually happens under hypoxic or low oxygen conditions. When a tumor is starving for oxygen, it packs its exosomes with a different enzyme, MMP-13.
Oh, wow. So it builds its own malicious demolition crew.
Exactly. These MMP-13-loaded exosomes travel through the bloodstream to remote organs, like the lungs or liver, and start degrading the ECM there to make room for invading cancer cells.
We usually think of metastasis as cancer cells randomly floating away and landing somewhere, just hoping they survive.
Right, but this means the tumor is actively sending out scouts and saboteurs to prep a landing zone in advance.
But it also promotes tumor angiogenesis, right?
Yes, the degradation of the remote matrix actually triggers the creation of new blood vessels, so the blood supply is waiting for the cancer cells when they arrive.
It is literally remote-controlled terraforming. That is terrifying, honestly.
It is daunting, yes. But understanding this hack is huge for medical science. Because if cancer can use these messengers, human scientists can engineer them to fight back.
Right, which brings us to the future of all this. Diagnostics and engineered therapeutics.
Let us start with diagnostics. Because of that lipid bilayer we talked about, Exosomes are incredibly stable in bodily fluids.
So they are perfect non-invasive biomarkers.
Exactly. We can detect diseases early or intercept those tumor scouts just by analyzing exosomes in a simple saliva, urine, or blood test.
No surgical biopsies required.
Right. But the therapeutics part is even more exciting. The sources give a prime example regarding urethral stricture, which is a severe scarring of the urethra.
And the culprits there are M2 macrophages, right?
Yes. In that inflammatory environment, those immune cells secrete exosomes carrying a microRNA called miR34A5P, and that actually makes the urethral scarring worse.
Okay, so how are scientists fighting back against that?
They are engineering stem cell exosomes. They stimulate the stem cells in a lab with TNF-alpha, an inflammatory cytokine.
So they stress the cells out on purpose.
Exactly. It forces the stem cell into a defensive posture. It starts producing defensive exosomes carrying a different microRNA, miR146A.
And what does miR146A do?
It completely suppresses the scar-forming fibroblast activation.
Wow. And they're even combining these engineered exosomes with physical scaffolds, right?
Yes, collagen nanoyarn scaffolds. They seed the scaffold with the exosomes, place it in the defect, and the tissue repairs itself with zero scar formation.
I mean, just think about the immense advantage this has over traditional stem cell therapy. So you get all the healing power of stem cells, but without the risks of immune rejection, or forming tumors, or trying to keep live cells alive during a transplant.
Exactly, because you are just using the instruction manual they leave behind. It is cell-free regenerative medicine.
Which is just a massive paradigm shift. So to recap this whole journey, we started by looking at this hitter exosome internet in our bodies.
Right. We saw them acting as foremen, handing out blueprints.
Then we saw them command demolition crews in the mouth to clear fibrosis, and we saw them deploy riot police in the joints to stop cartilage breakdown.
We saw how tumors hack the network to terraform distant organs.
And finally, how scientists are engineering these tiny armored trucks to heal tissue without scars. It is incredible.
It really is. And, looking at all this data leaves me with one really profound thought that builds on all this source material.
What is that?
Well, if our cells are constantly secreting these stable vesicles that are traveling through the blood and altering the gene expression of distant tissues in real time,
Yeah.
where does one organ end and another begin?
Oh, wow.
Right. Are we really a collection of distinct, separate organs, or are we just one giant fluid intelligence that is constantly rewriting its own biological software?
I mean, that completely shatters the idea that we are just static machines. You are this living, interconnected network. So for everyone listening, the next time you think about your body, take a moment to appreciate the billions of flash drives uploading instructions right now to keep you intact. Keep questioning the microscopic universe inside you, and we will catch you on the next deep dive.